Adam Rutherford, A Brief History of Everyone Who Ever Lived, The Stories in Our Genes. Embark on a journey through the fascinating world of our genes, as we explore the groundbreaking book, A Brief History of Everyone Who Ever Lived, The Stories in Our Genes by Adam Rutherford. Delve into a new understanding of human history and evolution through the use of paleogenetics. Uncover how our DNA holds the key to unlocking our past, from our ancestors' inner species mingling to the cultural and environmental adaptations that shaped us. Get ready to challenge your preconceived notions of human history as you gain astonishing insight into the complexities of genetics and how they impact our understanding of race and culture.
Unraveling our genetic past. While history books may convey certainty, the truth is that our knowledge of human history becomes increasingly uncertain the further back in time we go. However, the development of genetic analysis has provided us with a clearer picture of humanity's ancient past. Groundbreaking scientists such as Gregor Mendel, Francis Crick, and James Watson paved the way for the Human Genome Project, which successfully mapped the full set of human DNA in 2000. This progress led to the emergence of paleogenetics, a field that uses DNA to delve into the history of human evolution. Before Homo sapiens, or wise man, there were various other Homo species such as Homo neanderthalensis, Homo habilis, and Homo erectus. Homo erectus, one of the early upright apes, evolved roughly 1.9 million years ago in Africa and spread across the globe. Meanwhile, Homo sapiens likely emerged around 200,000 years ago in eastern Africa. As Homo sapiens migrated out of Africa into Eurasia, they encountered another human species, the Neanderthals. Remarkably, the initial meetings between our ancestors and Neanderthals were harmonious, with evidence of substantial interbreeding between the two species. Genetic analysis has revealed that the average European shares about 2.7% of their genetics with Neanderthals. Although Neanderthals have technically died out as a separate species, their genetic legacy remains alive within us. Through paleogenetics and the fascinating revelations it offers, we can now appreciate the interconnectedness of our deep evolutionary past. Milk and the culture connection. Genetic analysis not only provides insights into human evolution, but also sheds light on cultural developments. A prime example is lactose intolerance among adults, which varies across populations. A single gene variation leading to lactase persistence in Europeans, likely resulting from dairy farming, highlights the interplay between culture, genes, and environment. Similarly, genetic analysis allows us to track human migration and changes in physical characteristics, such as skin color, that occur due to shifting environmental demands. Cracking the code of human evolution has never been more accessible, thanks to our advances in genetic analysis. But did you know that these same tools unravel the mysteries of cultural development as well? Genes and culture maintain an intricate dance, influencing one another in ways that leave their imprints deep within our DNA. Take milk, for example. While some people enjoy it daily, a majority of adults worldwide experience lactose intolerance. So, what makes that small group of individuals capable of digesting dairy? The answer is hidden within the LCT gene, responsible for producing the enzyme lactase. Though present in all humans, the LCT gene typically becomes inactive after infancy, except among those of European descent. This lactase persistence traces back to a single gene alteration in Europeans, thought to have emerged between 5000 and 10,000 BCE, coinciding with the establishment of dairy farming. Genetic analysis suggests its origin lies around present-day Slovakia, Poland, or Hungary. Africa and Asia also saw the rise of lactase persistence through different mutations, supporting the idea that cultural practices contribute to human genetic adaptations. But it doesn't stop with culture. Genetic analysis allows us to chart the course of human migration and observe the changes resulting from adaptation to new environments. Let's look at Europeans again, specifically, their skin color. While early human migrants to Europe had dark skin for optimal sun protection, modern Europeans boast diverse complexions. This evolution, too, has been documented through genetic analysis. For instance, examining bones from 7,700 years ago in present-day Sweden revealed genes responsible for producing lighter skin, blonde hair, and blue eyes. In Decrypting the Human Genome, we unveil fascinating tales of the interplay between genes, culture, and environment, ultimately painting a richer picture of our collective past. Unraveling Native American Roots. Contrary to popular belief, Columbus wasn't the first outsider in the Americas, as Viking warriors already set foot on the continent 500 years earlier. Genetic analysis shows that people from Siberian Asia crossed the Bering Strait using an ice bridge between 29,000 BCE and 14,000 BCE. All Native Americans, spread across the continent, connect genetically to Greenland's Inuit people. Interestingly, the Inuit FADS genes, responsible for transforming fatty acids in fish, are found in all indigenous peoples of the Americas. However, using DNA to identify specific tribal affiliations is misleading, as American tribes intermixed with each other and settlers from Europe, resulting in no pure tribal lineage. Unraveling our noble ancestry. You might be amazed to learn that every person can trace their roots back to noble ancestry. Thanks to a mathematical model by Yale University statistician Joseph Chang, we now know that every European shares a common ancestor within the last 600 years, around the time of Richard II of England. This means that people with European ancestry are descendants of the 9th century King Charlemagne, as he fathered 18 children. Similarly, those with Asian or African roots can trace their lineage back to Genghis Khan or Nefertiti, respectively. While it's fascinating to discover our royal connections, it's also a good thing that not all our ancestors were royalty, as inbreeding within royal families often led to serious genetic disorders. The mind-boggling fact that we are all descendants of historical figures like Charlemagne, Genghis Khan, or Nefertiti may initially seem impossible, but it's actually a result of mathematics. As each person has 2 parents, 4 grandparents, 8 great-grandparents, and so on, the number of potential ancestors can quickly skyrocket. Yet, 1,000 years ago, there weren't billions of Europeans, Asians, or Africans. The explanation lies in the reality that many individuals in our genealogy fill multiple positions within any given family tree. Consequently, our lineage reaches back to nearly every person living in a certain period, royalty or otherwise. Nonetheless, we can consider ourselves fortunate that not all our forebears were royal. Inbreeding often occurred within royalty due to the practice of marrying within their own ranks, resulting in fewer ancestors and an increased risk of genetic diseases. A striking example is Charles II of Spain, who suffered from a severe genetic disorder due to inbreeding. Born with malformed genitals, he was unable to have children. While most people, on average, have 254 ancestors spanning 8 generations, Charles II only had a mere 82. So, as you ponder your ancestry, remember that we are all connected to one another through an intricate web of genealogy, with traces of nobility embedded within our family trees. Debunking genetic basis of race. Racial classification within genetics has long been a controversial topic. Early scientists like Johann Blumenbach attempted to categorize humans into 5 racial groups: Caucasian, Mongolian, Ethiopian, Malayan, and Native American. However, a study published in 2002 by Stanford scientist Noah Rosenberg demonstrated that race could not be easily defined through genetics. Through this study, he gathered genetic samples from over 1,000 people across 52 regions, and used a computer program to analyze the genetic similarities. The results showed that classification of race was arbitrary and that grouping people into categories based on nominal differences was baseless. In fact, the greatest genetic differences were discovered within racial groups, rather than between them. This evidence undeniably proves that our perception of race is deceiving and does not accurately reflect significant genetic distinctions. Unraveling the Human Genome. On June 26, 2000, President Bill Clinton announced a groundbreaking achievement, the complete decoding of the human genome, led by the Human Genome Project. This colossal scientific endeavor, which took 8 years to complete, unveiled three significant discoveries. Firstly, humans possess far fewer genes than initially thought, only around 20,000, less than roundworms and bananas. Secondly, the majority of our DNA, about 98%, is considered junk DNA, with little to no function as of now. Lastly, the complex interactions between genes debunk the previous belief that a single gene could cause specific outcomes, such as certain cancers. Instead, genome-wide association studies, GWAS, revealed that numerous genes contribute to medical conditions, working together to create cumulative effects that can result in illness. These groundbreaking findings have generated new perspectives on human genetics and a deeper understanding of our biological complexities. Debunking single-gene myths. The notion of a single gene dictating specific behaviors is an outdated and misguided understanding. The case of Davis Bradley Waldrop, who faced execution for homicide and used the MAOA gene variation as his defense, shows how misleading this idea can be. Genetics is much more complex, as no single gene can account for specific behaviors. Furthermore, epigenetics, or the inheritance of acquired traits, reveals that traits can be passed down through generations in some instances, yet still do not contradict evolution by natural selection. It is crucial to understand that genetics is an intricate field, and attributing responsibility to single genes is unscientific and lacks nuance. Dispelling the misconception that a single gene can control or cause specific behaviors, studies have shown that such claims lack scientific accuracy. For example, Davis Bradley Waldrop, who faced execution for homicide after committing a heinous crime, managed to evade the death sentence on the grounds that he had a variation in the MAOA gene. This gene is known to be associated with increased aggression, impulsiveness, and criminal behavior. However, using a single gene's variation to pinpoint the cause for one's actions is a misleading and overly simplistic argument. Contrary to the single gene fallacy, genetics has displayed its complexity and malleability through another intriguing aspect called epigenetics. The study of epigenetics posits that acquired traits can sometimes be inherited by the next generation. For instance, the descendants of survivors from the Hunger Winter, a period of extreme starvation in 1944, showed a higher prevalence of medical problems such as obesity and diabetes. This phenomenon doesn't challenge the concept of evolution by natural selection, but rather highlights the rareness and temporary nature of these inherited traits that may disappear after a couple of generations. In conclusion, creating a narrative centered around a single gene is a disservice to our understanding of the vast and intricate field of genetics. Epigenetics and the multigenerational inheritance of traits further demonstrate the complexity of heredity. It is vital to embrace a more nuanced and informed perspective on genetics, transcending misleading and oversimplified concepts. Evolution beyond natural selection. Just like the mutants in science fiction movies, humans have developed extraordinary abilities, but not through physical mutations. Instead, we've invented technologies like airplanes and night vision goggles. Human evolution doesn't stop there. As we continue to evolve genetically with every new baby, our DNA transitioning between generations. However, some genetic changes have proven detrimental, making protein production less efficient or dysfunctional. Natural selection has slowed due to modern medicine curing conditions that previously led to death, but it hasn't stopped entirely. As humans keep reproducing and dying, evolution continues to shape our species, with Darwin's theory remaining the best explanation available.
This unforgettable exploration of a brief history of everyone who ever lived. The Stories in Our Genes brings a wealth of groundbreaking knowledge about our past, present, and future as a species. Rutherford deconstructs human history and evolution through the lens of paleogenetics, providing a clearer understanding of how we're linked to our ancient ancestors and Neanderthal relatives. Moreover, the field of genetics unravels the links between culture, environment, adaptation, and our genes, revealing how our DNA helps us uncover the migrations and connections of our ancestors. As for the complex topic of race, genetic studies disprove any scientific basis for dividing humanity into racial categories. As we continue to evolve, we must remember that Darwin's theory of evolution is still our most valuable guide to understanding the dynamic nature of our existence.